Day 7. Day 7. We keep pushing it. We're trying to feed knowledge into here with the AI help. This was very important last time. The ancient retroviruses that did manage to infect egg to sperm cells millions of years ago, and their DNA is still in your genome today. About 8% of human DNA is fossilized retrovirus sequence. That's a very interesting. So we have repositories of knowledge, and some of that knowledge never used to build our parts, to build any part of our body. I had no idea about that. But let's move on. Let's move on. 1-1. Okay, question 22. Different territory. What is ATP and why does every biology answer seems to involve it? I mean, we learn a lot about it already. ATP, it's a highly charged molecule that's produced by mitochondria, and this is pretty much a energy currency. Majority of processes in our cells require ATP, and specifically the DNA replication as most important one, so our cells can divide. I should get 1 for that. We'll see. Clean and accurate. Yes. Finally. Is this one? By the way, I don't know how big this context already. No, still not the one. Oh, highly charged molecular, correct. It stores energy in a chemical bonds produced by mitochondria, correct. Primarily, also produced in small amounts by glycolysis, which happens outside of mitochondria. I think this is for anaerobic perspiration when they produce 2 ATP. Maybe wrong. Energy currency, correct. This is a standard term biologists use. Majority of processes require ATP, correct. DNA replication require correct. The quick detail how it actually works since you're a programmer. Let's see, ATP stands for adenosine triphosphate. The three phosphate groups. When the cell needs energy, it snaps off one phosphate group, ATP, become ADP. Adenosine diphosphate, 2 phosphates. That snapping release energy, which powers whatever process needed. Then mitochondria recharge ADP back into ATP by reattaching the 3rd phosphate. Right. Again, I'm gonna bring this over and over. That's what I need a t-shirt. Remember Civ 5? When Civ 5 introduced like 3 charges, or Civ 6 maybe, Civilization 6, the game, it introduced 3 charges on worker and then worker die. For some reason it remind me that. How much of it you use? Your body contains only about 250 grams of ATP at any moment. Really, but you recycle your entire body weight in ATP every single day. Recycle your entire body weight in ATP every single day. Let's see. So let's see how many. Okay, I'm so confused. It gave me that since you're a programmer, this is how it works, but it didn't give me anything. From this sentence, I understand following. Calculator, let's say my weight is 165 pounds. Since they're in gram, we're gonna do kilograms, sorry. 75,000 grams divided by 250, 300 cycles. So ATP converts to ADP and back to ATP 300 times per day. Recycle your entire body weight of ATP every single day. Each ATP molecule gets recharged and discharged hundreds of times daily, 300 to be exact. We just calculated. Like, any moment by your recycle your time daily. The throughput is staggering. Really? Why every answer in the valve with it? Because it's the universal adapter. Doesn't matter if the cell is dividing, contracting a muscle, firing a neuron, pumping ion, building a protein. The energy payment is always in ATP. It's a single currency the whole cell economy runs on, like every microservice in the system using the same message format. I would say it's similar like for gas, probably in Ethereum. Let's compare it for shitting and giggles, I guess. Is it correct to compare ATP energy currency for cellular biology to the gas in the Ethereum cryptocurrency network / framework? It's not a network and it's not framework, but the word cryptocurrency, Ethereum cryptocurrency, blockchain. And yes, it holds up surprisingly well. Let me push on it a bit. So show where it really fits and where it's break. I would guess that it breaks because it doesn't. It's not a cycle, right? You need gas, you spend it, and the gas, like in ATP and DP, it's a cycle. ADP, ATP, ADP, ATP. And mitochondria put it back. And gas is just kind of one way. You pay, you put transaction in your chain, and you move on. Units of doing work, you don't execute operation for free, you pay per operation in standardized unit. Yes, the unit is decoupled from what's being done. True, you can pay gas to transfer token, call a contract, or store data, just as you can spend ATP to contract muscles, pump iron, blah, 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 blah. The picture structure maps well to different EVM opcodes cost different amount of gas. Different cellular operation cost different amount of ATP. Translation: one amino acids on the growing peptide chain cost 4 ATP. Equivalent pumping Na plus ion cost a fraction of 1. Synthesize a fatty acids cost a bunch. Interesting. In both systems, the cost roughly tracks the thermodynamic computational difficulty of the operation. Yep. And crucially, in both systems, the currency gets consumed and regenerated in cycle. Oh, I was wrong about this. So the currency actually regenerated in cycle. I thought compute is not regenerated. Gases paid, burned, or given validators, or given to validators, and users top up the ISP balance. Yes, that's not the cycle. What cycle is this? I guess if we are mitochondria and we just put in money on ETH balance, ATP is hydrolyzed to ADP plus pi, then regenerate back to ATP by mitochondria or glycolysis or substrate level phosphorylation. The ADP-ATP ratio is roughly analogous to how much gas do I have left in my wallet. When it breaks, the analogy gets wobbly in a few places. Gas is particular mechanism. ATP is actual thermodynamic substance, yes. Here's the pricing mechanism, and this one is actually thermodynamic. It's a truth of physics. Gas exists to allocate scarce block space and prevent spam. It's an economic construct. ATP exists because phosphorite-bound hydrolysis release 30 kilojoule per mole of free energy that can be coupled to otherwise unfavorable reaction. That's interesting. So what the gas in Ethereum invented as an economic construct to prevent spam and etc. and give some basic monetization options, right? But ATP is a physical, so specifically it's release energy to do unfavorable reaction. So my question now is, what in compute or in software, what in compute or software could be released energy that can be coupled to otherwise unfavorable operations. Slash transactions. That's interesting, like, because sorting comes to mind. Great question. You're probing the part where the analogy breaks, which is exactly where the interesting physics live. Ooh, let's go. Let me try to find out find you a real mapping. The things you're looking for is entropy information, and the mechanism is Landauer principle. Oh, let's go. Let's go. We're going wide today. A physical principle pertaining to a lower theoretical limit of energy consumption of computation. All right. So in nature, everything want to fall down to the lowest energy. I'm blanking on the word. Energy band. Let's go into the images. Yeah, this one, like, it will not stay here, it will just fall here. Stage 3. And we can push it up and it will fall. Interesting. So, is there a way we can, okay, Twitter message on X for day 7? Is there a way we can use ATP as an energy in body to help training MLs to avoid local minimums? Obviously, I didn't write it down. I'm gonna do it one more time. Is there a way in ML training we can use something similar like ATP in the biology, cellular biology, to avoid local minimum traps? Please formulate that question in better grammar so people uh, would understand what I mean. And we'll get back to it. Meanwhile, so we went to this one because when I see this, I right away see local minimums. And when you train any model, that's what you're hitting, right? This is cool. Detection. Oh. Is this the one how we can split music into different instruments? Obviously not. Initiate state, global mass, quench, full state recognition. And your approach to probing Landor principle in the quantum many body regiment. This is so cool. Like, who is working on this? I want to be part of it. Our interest centered on how concepts such as information deletion and heat production might manifest in this uniquely tunable quantum regiment, said Isert. This naturally brought us to Landor principle. I love this. Too distracted to read all this, but I will. Energy per bit of operation, reversible computation. Here's the things like computation cannot be reversible, right? That's why. But what if we can pack computation into the software itself? What if software can unpack and compute something? That's crazy and fascinating. It's fascinating if it is the answer to that. It's crazy because it's fundamentally not how the software works. Software will not work without compute. I'm going away from biology, but this is interesting. So landscape. Here's the physical fact: erasing one bit of information in computer has a minimum thermodynamic cost in joules in heat dissipated to environment. No, it's taken it from a hardware perspective. I'm trying to approach from different angle. This isn't an engineering limit, it's a law of physics derived from a second law of thermodynamics Rolf Lander provided in 1961. I'm not trying to break a law of physics or thermodynamics. Obviously, that's a rules of the simulation. But I'm trying to approach it from different perspective. The flip side, which is what you are actually asking about, a system in a known low entropy state can be used to derive an otherwise unfavorable operation. Yes, that's exactly what I was asking earlier. Like in the bottom, I asked about ATP. Yes, that gets spent in the process by becoming randomized. And gets spent in process of becoming randomized. Yes, I think sorting. Sorting should be the one. This is the real parallel of ATP. Let me draw it out. ATP sites have computation, a register full of known zeros, or any known bit patterns is a low entropy configuration. The system wants to randomize. Increase entropy is thermodynamically favorable. You can couple that favorable randomization to an unfavorable computational operation and extract work from the bits becoming random. Yes. Yes. We need to extract the work from the bits becoming random. Imagine. That would be so cool. John Lennon. Imagine, I wonder if you can. You get the computations that otherwise would have required energy input. I love this. Text edit. We're gonna do new, we're gonna call it save. And baby we go. And you can couple that favorable randomization to an unfavorable computation operation and extract work from the bits becoming random. I love this. The clean bits get spent, you get the computations that otherwise would have required energy input. Oh, I know. Is the e-ink something similar because it's require energy only when to flip the bit from black to white or from white to black, and then it just stay without power for ages? The concrete demonstration of this Maxwell-Demont experiments. Physicist actually has built system. Toyable Bernoulli, where information about the particle state is used to extract work from thermal fluctuation. The information that gets consumed, the memory gets randomized, and mechanical work comes out. The information is literally fuel. Whoa. What? What? Ooh, not this. Oh, I keep forgetting to reply. Did they shark a cat again? Why is this devil? my god, stop it. Physics. Come on. Demon. Continue. Start a sentence. Like, this is all cool. Like, I want to know and understand all this. But we'll skip it for now. ATP, clean beats, blah, 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 blah. So let's see. Here's the cleanest information of your question. In ML training, is a technique analogous to ATP-driven reactions in biology where separate energy source is spent to push the system through thermodynamic unfavorable transaction that could help optimize escape local minima. Nah. Like, if I read this, it's like, what are you talking about? Can we borrow the ATP trick from biology, coupling an unfavorable steps to favorable energy release to help gradient descent escape local minima? That's better. I love this. And to actually answer it, yes. Whoa. Maybe someone already done it. And the connection is remarkably direct. Your intuition is pointing straight at real family of methods. So many compliments. I know they fake. I know they generated, but they sounded so nice. Oh. The core idea in biology is that thermal fluctuation alone aren't enough to climb energy barriers on a useful time scale. So cell inject energy ATP to driven transitions that wouldn't happen spontaneously. The ML analog is almost the same picture. Vanilla gradient descent can't climb out of local minimum because by definition the gradient points back in. You need to inject something, noise, momentum, or both to push the system over the barrier. Damn right. The most literal version of this analogy is simulated annealing, and it's a ML cuisine stochastic gradient Langevin dynamics. Okay, we found two cousins. On the left side we have SA, and the right side we have SGLD. What the fuck is this? Oh, by the way, happy Earth Day. Probabilistic meta-res algorithm used to find the global optimum of function, particularly in discrete search space with many local minimums. That's what I was yeah. We have many global minimums. We start from this with gradient descent, we'll fall here and never go back. So we'd have to step up. Bam, bam, bam. Yes. Okay. I tried this before. Where the analogy gets really sharp. Oh, wait, what did you? The noise plays a role in the interactivity biology momentum and Adam, which you almost certainly used. And Adam, which you have almost certainly used. I mean, is a software version of the same idea. Momentum accumulates a velocity vector across. Oh, yeah. You basically decrease or increase the step. So when you hit a local minimum, you don't stop, you coast through it like a ball rolling down a hill with inertia. The stored momentum is functionally a small ATP like reverse energy banked from previous favorable steps. That's good. Yes, right. So if you do something that's in line with intent, you can store that energy as momentum. I like that. I like that. And then you can apply that momentum by a concept, by a construct, right? It's still software, it's still a contract, but you apply it to the unfavorable. And maybe your false boolean will switch to true. Interesting. It's hard for me to explain this using proper words and labels and sentences. But you see where I'm going with it, right? The stored momentum is functional as small ATP-like reserve energy bank from previous favorable. Yes. Where the analogy gets really sharp is in a method that explicitly allocates the energy budget. If you works known. Cyclic learning rate, warm restarts. Periodically crank the learning rate back up in mid-training. So it's yeah, I see. This is the most ATP-like move in practical ML. You're saying we have settled into basin, by basin. Yeah. Now inject fresh energy to see if there's a better one nearby. The schedule is the metabolism. Replica exchange. Parallel tempering runs multiply copies. These different approaches to find the local minimum and a global minimum, and not to misinterpret the local minimum for the global optima, right? A little bit different, but interesting. ATP analogy is slightly off in one respect. In biology, ATP is spent on specific reaction via enzyme coupling. Its target triggered in most ML optimizers. The injected noise energy is undirected. It's just randomness, hoping some of it lands on productive direction. Yes, we just hope it will. We will find it. So to answer this question, yes, and we already do. We just don't call it that. I really like this one. I really like this one. How to explain it? Imagine you have hard drive, right? That stores 0 and 1s. And it uses same energy to store either 0 or store either 1. They identical. But what if zeros was harder to store? And by nature, it was always want to fall into ones. Or opposite. Ones is harder to store and it always want to reset into zeros. Therefore, from 1 to 0, it's favorable, and we don't have to compute, it will compute it by itself eventually. But from 0 to 1, that would need the ATP. Is that the way how atomic clock works? Probably not. I think we should go back to biology, but I love this. This energy, I wanna try, like I wanna see how we can. I want to see how we can use concept of ATP ADP cycle in. So I think I'm cycle software. That's what AI does to you. Like software engineer doesn't know how to spell software anymore. Welcome to 2026. So I'm thinking it a little bit wrong. I'm trying to use compute as the energy in a software, but software unaware of compute, right? Software runs by compute, but it's not aware of compute. That means I'm bringing concept outside of box into the box, and that should not happen. It will never work. But what can we do? We maybe can apply that for some kind of smart caching mechanism, right? When software saying like go and save to the database or to the file, and maybe it's need quote unquote energy that it can produce from the gets. Okay, I'm gonna try one more thing, sorry. I think I started thinking about it in a wrong way. I was thinking in a software analogy, but for energy, I was bringing compute, but compute is outside of software. Software cannot run without compute. So that was wrong. What if we still sticking with software and trying to recreate a cycle of ATP into ADP and then back to ATP with the concepts only inside of the software? And what if we will use a get as favorable transaction in a caching mechanism versus post that's unfavorable? So post will write into cache and get just get it, or maybe other ways around. In other words, can we find analogy with the smart caching mechanism? You caught your own category errors cleanly. Let's remember that. I coded cleanly, like fish in a pond. And the reframe is good. Let me engage with the caching idea directly, because I think it's closer, but not quite the right mapping, and the right mapping is interesting. Why the get post framing doesn't quite work? The key property of ATP isn't that one operation is cheap and the other expensive, is that the expensive operation pre-built are reusable high potential resources that many different downstream operations can draw from to do otherwise impossible work. Exactly. ATP is fungible. One ATP molecule can power a ribosome, a muscle, or an ion pump. Yeah, we heard that 3 times already. It's decoupled energy store. In your get post caching analogy, the post builds a cache entry and the get reads. Yes, that analogy was wrong. I admit it. I was just trying to start conversation. But the cache entry only powers that specific query. It's not fungible. It's more like pre-computing a specific answer that's storing generic energy. A cache hit save works on exactly the computation that was cached, not on arbitrary other computation. So the analogy captures pay once, benefit later, but misses the fungible currencies, part that makes ATP special. The closer analogy, memorizing it isn't quite easy, but it points the right way. Memoization has the same limitation. It's tied to specific function argument pairs. What you want is something that stores generalized potentials that can be drawn down by many different consumers. Okay, is it a map? Where the analogy actually lives, connection pools, token buckets, and pre-computed indexes. Very interesting. Connection pools. That's very interesting. So because we not always know how big the pool should be, right? And if the connection pool is expensive, optimizing it, that would be a good idea. Token buckets. That's interesting too, and pre-computed indexes. I'm not really advanced in indexes, to be honest with you, so maybe. Here's the 3 software patterns that are much closer to ATP cycles, increasing order of how well they fit. OpenATP connection or database connection is expensive. Handshake, AUS, TLS negotiation, yes. A pool does this work ahead of time and keep end connection charged up and ready. Any request that comes in can grab a ready connection, do its work, and return the connection to the pool to be reused. The expensive setup is decoupled from the cheap use. Connection gets spent, used for a query, and regenerated. Return to a pool, possibly health checked. This is a generally good analogy. Pool is your ADP ATP ratio, and a background worker replenishes exhausted connections the way mitochondria replenish ATP. The one that I think really nails is speculative execution and branch prediction. This might be the best software knowledge, and it's a weird one. Modern CPUs speculatively execute code down branches before they know whether the branch will be taken. They spend energy, literally, transistor switches to pre-compute the results that might be needed. When the speculation is right, the expensive upfront work makes the real computation effectively free. When it's wrong, the work is discarded as heat. Are we talking about compilers? and agents. But here's a deeper things your question is circling. I think what you're really asking is, yeah, go, tell me, can software have a genuinely internal energy economy without reaching outside to compute? Yes. Given we have a constant compute, like we have a sun and constant energy beam, right, going to our planet. Its changes is vary based on season, rotations, etc. But given we have that as a constant, given we run in a simulation, a compute constantly, maybe with some variation, can inside of that simulation, inside of software, we have a genuinely internal energy economy? Besides the entropy that everything want to fall apart. And the answer only in a limited bookkeeping sense. Software can simulate energy economies, token buckets, connection pool, especially cache, but there is nothing to pure software that force the asymmetry the way thermodynamics force ATP asymmetry in a cell. I like this. The one place this does become real is when the software economy is enforced by an external scale resource, database connection, API rate limits for Amazon, for example, GPU memory, network bandwidth, then the scarcity is genuine and the economy has teeth. Connection pools, ATP, like, so caching isn't right. Okay, let me go back to the questions. I'm very disappointed that I only get 0.9. 22. What are stem cells and why are they special? I needed 2 minutes break. I'm not gonna look for the stem cell. There is no point. When you learn something, there is no point to cheat. Be back. Okay, I'm back, and I have a million questions, of course. Number one is so basic, and we need to create like every 6 days I'm gonna clear tabs and start from scratch because this is unmanageable. Is plants have mitochondria? Yes, plants cell have mitochondria, while chloroplasts produce food via photosynthesis. Mitochondria are essential for cellular respiration, breaking down sugar. Oh, right. Right. So I'm gonna eat a bunch of mitochondria inside of the cells. What are the stem cells and why are they special? My current understanding on a day 7 of study that stem cells are basic cells or based, I put it base cell like in programming, base, that not specify in any particular function or any not become specific yet. So it's not a muscle, it's not a neuron, it's not skin, it's a base. And then by epigenetics, I believe, or by other environment, depends what other cells surrounded this one, the surround the stem cells, it will become one of them. It will pick a function and become that cell. Second part might be off, but I was trying to bring analogy from the stem shots, right? Like if you pull the muscle, not a pull, like break the muscle and you do a stem shot into the muscle, it will regrow. I mean, it's not exactly that, but in basic analogy, that's what's up. Unspecified cell not yet committed to a function. Correct. Biologists call this being undifferential. Undifferentiated. Can become different cell type. Correct. This ability is called potency. Environmental factors surrounding cell determine what they become. Correct. So not epigenetic. The signals from neighboring cells, chemical gradients, direct cell-to-cell contact, tells the stem cell which genes to turn on and which to silence, pushing it towards a specific fate. Interesting. What if you're in a boundary, like between muscle and skin? Through epigenetic, you actually right here and more precisely than you might realize. Differentiation is largely in epigenetic processes. Every cell has the same DNA, but as a stem cell comes to become, say, a neuron, epigenetic marks permanently silence muscle gene, skin gene, liver genes, etc., leaving only neuron relevant gene accessible. Interesting. So that's a process. The DNA doesn't change. Yeah, I know the DNA doesn't change. The reading permissions change. So your instinct to connect stem cells to epigenetic is correct. The base class analogy. Extended. The level of potency maps nicely to inheritance depths. It's the levels of potency. Totipotent, the fertilized eggs and its first few divisions can become anything, including the placenta, the ultimate base class. No inheritance interfaces implemented yet. Sorry, not an interface implemented. Pluripotent embryon cells, stem cells, can become any cell type in the body, but not the placenta. By the way, placenta from retroviruses. I didn't know that. Multipartent adult stem cells can become several related types, but not anything. A blood stem cell can become red blood cells, white blood cells, platelets, but never a neuron. Right. Uniparent can only produce one cell type. Okay, next. Probably eating during the stream not a good idea, but I'm starving. I need some sugar to go into my mitochondria and create some ATPs, so my neurons can fire and remember all this. By the way, we still don't know how the memory works, right? So neurons fire. We know that it's happened during the sleep, replaying backwards and writing it, but then writing it into what? Not the microtubules, right? We don't know 100%, right? The core idea is synaptics plasticity. So the first idea was in 1970s that it's memory stored in how strong the connection is. The connections between neurons get stronger or weaker based on activity. I don't think that's what's up. Especially now, knowing AI, MLs, artificial neural networks. The connections are not memory. They basically statistical probability outcome. I mean, in software, right? In real brain, I have no idea, but it feels like it's not memory. Yes, that's what it's saying. The molecular cascade of LTP, the well understood part. In the classic hippocampal LTP story, NMDA receptors. long-term potentiation. See, I was wrong. It is that how it's stored. Let's get harder. The persistence problem. Here's the things that should bother you as a programmer. Proteins turnover. Individual AMP receptors get recycled in hours. KMK2, CAMK2 molecules degrade, yet memories last decades. How does a pattern persist when its substance is constantly being replaced? This arguably, if it replaced with exactly the same stuff, then like you have a ship, right? The old ship from 1956. And every day you replace one wooden piece of it because it just degraded. After 50 years, you replace the whole ship. The all details, all parts are new, but it's still the same ship. I mean, is it the same? The shape and everything is exactly the same. Material is different. Oh. Am I so statistical? That's like I explaining exactly what the AI gives me? This is the ship of treasure problem of memory. A genuinely unresolved, if you count it as mechanism. Is the ships of treasure exactly what I just explained? Yeah. Damn. Yeah, you replace every single one. That's cool. Epigenetic marks. So where's the self-sustaining kinases activity? Prior like, oh, so that's what's memory. Are they prion like protein CPEB? Not sure. Structural changes, epigenetic marks. I don't think it's epigenetic marks, it's too small. Or maybe it is. We have a bell number, right? Maybe it is a bell. Like we don't remember everything. But then we can recover and recreate. So yeah, it's not epigenetic. It's not the structural changes. I think this one, or I'm putting my bets into this one. 2026, April 22, day 7. I think the memory stored by this option. From out of this four, I don't understand neither of them. Just slightly, maybe. But I'm putting $20 on this. The system level is a big gap. N-gram cell. Neurons that are actively during learning and whose reactivation can trigger recall. Yes, no, this one. Okay. I take my $20 from this and put in $100 to engram cells. Question 24. What does the liver do? I think the liver main function is to filter out different toxins from our bloodstreams. And those toxins could be either some byproduct, like for example, anaerobic respiration with a byproduct of lactic acid that's we done yesterday, or it could be even alcohol, right? That's we take externally. Yeah, I think liver acting like a big filter to filter out byproducts and stuff that we don't want to have in our blood. And also it does metabolize stuff. I don't know what exactly, but it does. I also think I saw somewhere recently in a couple last days that some immune cells, B cells or T cells, actually not located in the liver, but actually doing those triggers reaction or signaling. Majority of immune cells located in the lympho, not the channels, lympho sacs, I guess. Lympho chambers. Let's put them chambers. So for liver, it's filter out and metabolism. I named two. Solid core answer with a nice callback to anaerobic respiration lesson. Damn, that's how we do it. Filter toxin from the bloodstream, correct. The liver is the body's primary detoxification organ, correct. Alcohol metabolism, correct. The liver breaks down ethanol using specific enzyme, alcohol dehydrogenases. Yeah. This is why heavy drinkers destroy the liver first. Metabolizing things, correct. And this is actually the biggest story. Let me expand. Lactic acids as a byproduct, correct callback. The liver actually recycles lactic acid back into glue. Oh, really? No way. It takes a lot of energy. It takes a lot of ATPs. Call the Corey cycle. Nice connection. Immune cells in the lymph, not liver. Mostly correct. The lymphatic system and the lymph nodes are major immune hubs, though interestingly, the liver does contain resident population of immune cells, Kupffer cell, that filter bacteria coming from the gut. What you are missing? The liver is far more than a filter. Okay, the liver does an absorb number of jobs. Oh, sorry. The liver does an absorbed number of jobs. Okay, it's arguably the most multifunctional organ. I was so wrong. I will get like 0.3. Bile production makes bile. I have no idea what is bile, which is essential for digesting fats in the small intestine. Oh, in the small intestine. Bile makes bile. That produce liver. Bile is yellow greenish fluid produced by the liver stored in the gallbladder. Oh, желчь, right? Yeah, желчь.
Желчь. Bile.
Interesting. I didn't know liver produced bile. Glucose regulation. stores glucose glycogen when blood sugar is high, release it back when blood sugar drops. This major homeostasis loop. Call back to question 15. Thank you. Glucose regulation. Protein production. What? I thought protein makes by inside of the cells. Makes albumin, most abundant blood protein, clotting factors, and many other blood protein. So it does make some proteins for the blood. Interesting. Fat metabolism, processed lipids, makes cholesterol. I need to watch a couple of videos about it. Storage, warehouse vitamins A, D, B12, and iron. Had no idea. Detoxification. The filter function you described breaks down drugs, alcohol. The liver is less like a filter and more like a chemical processing plant that happens to also filter. It's performing hundreds of different chemical reactions simultaneously. Fun fact, the liver is one of the few organs that can regenerate. You can lose up to 70% of your liver and it will grow back for full. Wow. The ancient Greeks somehow knew this in the myth of Prometheus. An eagle eats his liver daily and it regrows overnight. Oh wow. 05. Totally different direction. What is a mutation and are all mutations bad? Mutations, it's a small differences in the DNA when cell divides. I think it happened in the regular human, it happened from 50 to 200 times. Some mutations are beneficial. That's how evolution works. It helps adaptation. That's it. Small difference in DNA that occur during cell division, correct? Mutations are changes in the nucleotide sequence. A letter gets swapped, deleted, or inserted, or a whole chunk gets duplicated or rearranged. Some are beneficial, that's how evolution works, correct? There's a critical insight most people miss. Mutations aren't inherently bad. They're the raw material for natural selection. Without mutations, there is no variation, and without variations, there is no evolution. 5200 times, close to real number. Each human cell division produces roughly 0.5 to 1 new mutation. Over your lifetime, your cell accumulates thousands, but per generation, parent to child. A newborn carries roughly 60 to 70 new mutations that neither parent had. So your order of magnitude is the right ballpark. Yeah, that's what I was saying, over the lifetime. The filler picture on outcomes. Mutation fall into 3 buckets, and the distribution matters. Natural, vast majority. Nothing functional, it's in a stretch of DNA that doesn't code for anything, or it swaps a codon for a synonym. Remember codon degeneracy from Q4? Yes. Multiple codon code for the same amino acids. Most mutations are invisible to organism. This is like changing a comment in your code. The binary doesn't change. Harmful, small minority. The mutation breaks a protein or disrupts the regulatory elements. Sickle cell anemia is a single letter change in one gene, one nucleotide. Cystic fibrosis, same thing. Okay. Beneficial, very rare. The mutation improves neutral. Okay, not natural, neutral. To improve fitness in the current environment. Lactose tolerance in adults is 1. A mutation that keeps the lactose gene turned on past childhood. Most humans are lactose intolerant. European and East African population evolved the mutation independently because dairy farming made it advantageous. The counterintuitive part. Some mutations are harmful and beneficial simultaneously. Sickle cell anemia, one copy of mutant genes gives you malaria resistance. Oh, interesting. Beneficial. 2 copies give you diseases harmful. Natural selection maintains both. Correct on the essential, missing the neutral category, which is actually the biggest one. And the specific mechanism. Half checkpoint, 25 down, 25. You're holding steady around 55%. Want to keep going. Let's do a time check. What are we? 1 hour in. Yeah, let's keep rolling. What is the difference between a tissue, an organ, and organ system? The tissue, it's a range of many same moleculars, same combination or in the same pattern or same type. Sorry, not moleculars. The tissue is a range of same cell types in a certain pattern. Brain tissue, muscle tissue, skin tissue. The organ, it's a cell arranged into functional, into thing that perform certain functions, like chemical factory, like liver we just discussed, or heart. So it might even have different cell types within same organ. And the organ system, it's systems that involve multiple organs, I guess, what we have in our body: heart, lungs, liver, kidneys. Tissue group of same cell type, correct. Muscles, tissue, nerves, tissue, connective tissue, epithelial tissue. The 4 main categories. Oh, actually, I know what I'm gonna ask now. Epithelial tissue is the primary bodily tissue that covers external skin. So it's skin, lines, internal cavities, organs, and forms glands. Okay, so it's kind of like membrane for organs. I would guess this is for stomach, but no, lungs. Interesting. Organ multiple tissue working together for a function, correct? The heart isn't just a muscle, it's a nerve tissue to keep rhythm, connectivity issue, wall structure support, and epithelial tissue lining the chambers, multiple tissue types, one function unit. Organ system, multiple organs working together, correct? Your organ system example, you listed heart, lungs, but those are actually from different system. Oh, right. An organ system is organs that collaborate on one job. Cardiovascular system, heart and blood vessel. Respiratory system, yes. Yeah, I remember that from biology. Totally ruined it. Urinary system, nervous system, yep. 74. Yeah. Organ system and then organism. What is the difference between mitosis and meiosis? Oh, it was mentioned somewhere above, and I genuinely do not remember. Something borrows somewhere a molecule or atom. I think it was mentioned somewhere above, but I don't remember what it is. Something that on a molecular level, some mechanisms that borrows atom or proton or something to do certain function. Yeah, I don't remember. Oh, cell division. Yes, completely different from what you are picturing. Mitosis cell divisions that produce two identical copies. Or completely different. Mitosis cell divisions that produce two identical copies of the original cell. Same numbers of chromosomes, same DNA. This is how your body grows, heals, wounds, and replace old cells. Every cell in your body, except sperm eggs, was made by mitosis. Think of it as a git clone exact copy. Not sure if it's a good example. Probably. Mitosis. Meiosis, cell divisions that produce four cells, each with half the chromosomes. This is exclusively for making sperm and egg cells. Gametes. Humans have 46 chromosomes, so meiosis produces cell with 23. When sperm 23 meets egg 23, you get a new organism with 46. So my question is, what if you already have 23, then meiosis happen and you have 12 and a half? Why meiosis matter beyond just having? During meiosis, two critical things happen that don't happen in mitosis. Crossing over, matching chromosomes from your mom, and that physically swap segments. Things shuffles gene into new combinations that never existed in either parent. Random assortment. Which of your mom's versus dad's chromosome goes into each gamete is random for each of the 23 pairs. This alone creates 2 in 23, 8.4 million possible combinations per gamete. Combine crossing over the random assortment of two parents and the numbers of possible unique children from the same two parents is astronomically large. This is why siblings look different from each other despite having the same parent. So I'm a little bit confused. So we have a regular cell, right? It perform meiosis, and 46 original chromosomes in that cells split into 4 cells, and each of them get 23. So before it's split in 46 become 92, then it's split into 4 cells, each of them getting 23, and now it's a sex cell. But then when sex cell split, does it go through the mitosis and copy itself identically? So it used to have 23 and become 23, or going through the meiosis one more time, but then it should have only 12 and a half. So what is the trick? Great question. This is exactly the kind of confused words tapping to resolve. The answer: mature sex cells, sperm, eggs, don't divide aging. Sorry, the answer is mature sex cell, sperm, and eggs don't divide again at all. Also, they divide it once and they stay. They are end of the line cell. Their only job is to find each other and fuse. Okay, here's a full sequence. Regular cell, 46 chromosome, mitosis, 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 building up a supply of precursor cells. One of those precursors enters meiosis. Meiosis 1, 46, 2 cells with 23 each. The halving step. Meiosis 2, each of those 23 chromosome cells split again, but this time it's more like mitosis. It splits the chromosomes without having to, without halving. So 2 cells, 4 cells, 23 each. Mitosis 2, each of those 23 chromosome cells splits again, but this time it's more like I think it's screwed up something here. Result: 4 gametes each with 23 done. They never divide again. Then sperm 23 plus egg 23. Yeah, that's classic. Osmosis, no idea. I don't know. No worries. That's the first question that I didn't even guess. Osmosis, the movements of water across the membrane. All right. From an area of low solute concentration to. I saw it yesterday in video. That's why you cannot. That's why you can die from this if you're gonna drink a lot of salt water because your cells will starve from the water because the outer water of the water outside of cells will be more salty than water inside of cells, right? Simply if area of low solute concentration to high solute concentration. That's it. The simplex example. If you put a cell in very salty water, water flows out of cell towards the highest salt concentration outside, and the cell shrivels. Put a cell in pure water, water flows in towards the highest solute concentration inside the cell, and the cell swell and can burst. Oh wow. The programming knowledge is like a load balancer, but for water. No, it's not. No, it's a one-way function. Water automatically moves to equalize concentration on both sides of the membrane. The membrane is selectively permeable. Water passes through freely, but the dissolved stuff, salt, sugar, proteins can't. So water is the only thing that can move to fix the imbalance. I'm confused now. Why does this matter? This is how your cells maintain their size and shape. How your kidneys filter blood. Whoa, what? How plant roots absorb water from soil and why drinking seawater kills you. It pulls water out of your cells. Yeah, call this. I think I'm done. Like, what does pH mean and why does it matter in biology? So it's either your cell or whatever, the environment alkalic, like, or acidic. Okay, let's do one. pH measure level of acidity or alkalinity. Like, if your environment is alkalic or acidic, meaning more sour or more, I don't know, antonym of that. Alkalinity. Correct and to the point. Measure alkalinity, acidity, alkali, correct. Acidic versus alkaline spectrum, correct. A bit more detail on a rounded. The pH scale runs from 0 to 14. 7 is neutral, pure water. Below 7 is acidic. Above 7 is alkaline, also called basic. The scale is logarithmic. Oh, that's I didn't know. pH 6 is 10 times more acidic than pH 7. 5 is 100 times more than acidic than pH 7. Why biology cares obsessively about pH? Proteins, specifically enzyme, only work within a narrow pH range. Shift the pH even slightly and the protein's 3D shape changes. It denatures and stops working. This is why your body maintains blood pH between 735 and 747 with extreme precision. That's homeostasis loop. Call back to question 15. Homeostasis loop. Go outside the trench and enzyme fails. Chemicals reaction stop, you die. Different environments in your body runs at different pH level. Stomach, pH 2, extremely acidic. Dissolves food, kills bacteria. Blood, pH 7.4. Slightly alkaline. How to pronounce this? Where's my translate?
Alkaline. Alkaline.
Slightly alkaline. Small intestines, pH 8. Wow. Alkaline. Neutralizing stomach acid. Yeah, makes sense. Each environment has enzyme turned to work at that specific pH Pepsin stomach enzyme works as pH2. Wow. And dices pH7. Trypsin intensins enzyme works at pH 8 and dices the same potential. Yeah. Okay. Day 7 successful.